In this video, we will discuss about the CTSK protease effectiveness on overall glycocalyx thickness. So far in our previous video, we have understood that CTSK protease has a specificity beyond the mucin one, and it can cleave off other glycoproteins, glycans also. So considering that observations or considering that findings, they wanted to check whether the CTSK protease is effective on the glycoproteins that are overexpressed on the cancer cells, so that it can help in the reductions of the glycocalyx around the cancer cells. So for that, they selected this polysialic acid, chondroitin sulfate, Hepran sulfate. And the idea is that even though the CTSK does not have a specificity towards these glycoproteins, if it can cleave the carrier proteins of these glycoproteins, it can help in the reductions of the thickness of the glycocalyx. And for the controls, what they have used is endo-Na for the polysialic acids, and this for the chondroitin sulfates they have used chondroitinase ABC, and for the heparin sulfate they have used heparinase 1. And they did the studies in the in vitro system where they took the different cell lines and treat them with this 77 nanomolar CTSK, and for 1 hour at 37 degree at pH 6.75, and thereafter they checked the markers for these three different glycoproteins, that is FGF, F2 for the heparin sulfate, anti-polysialic acid, and anti-chondroitin sulfate using the flow cytometer. The results for the heparin sulfates can be seen here, that they have used the 4 different cell lines H82, OVCAR3, MCF10A, and MCF10AMUC1, which is overexpression of the MUC1, that cell lines that they have developed, not MUC1, MCF10A MUC1. Yeah, so MUC1 is overexpressed in this MCF10A, these cell lines that they have developed, which work on doxycycline treatment, based on doxycycline treatment. So here we can see that in the CTSK treatment, for all the cases, there is a significant reductions in the FGF2 staining or heparin sulfate, which is comparable to the control, that is heparinase. And for the polysialic acids, and here the heat inactivated results shows no reductions in the FGF2 staining, indicating that the specificity of the CTSK only that cause the reductions in the heparin sulfate. And the results for the heat inactivated CTSK is not shown for MCF10A and MCF10AMUC1. And for the polysialic acids, they see a reductions in the H82, but not in the OVCAR3. This indicating that these H82's polysialic acids are more prone towards CTSK cleavage or CTSK protease activity, whereas OVCAR is not. OVCAR polysialic acids are less prone to this CTSK protease activity. And for the panel D, here they have shown the results only for this one cancer cell lines, which shows that the chondroitin sulfase also goes down for H82 significantly when they use CTSK, and which is comparable to the control, that is chondroitinase. And thereafter they check the viabilities. The viabilities are also not affected that much. However, in the case of H82, the viability increased significantly for the CTSK treatment. And however, the reasons that they have not provided much insight about this increased viability of the H82. And thereafter, here they are checking the individual glycoproteins, but finally in the final panel they have shown the results for the overall effect on this glycocalyx thickness, and they have checked it for the CTSK in this YSCCC cell line, which also expressed high MUC1 glycoproteins, and this MCF10A and their MCF10A overexpressed MUC1. And they can see that for the case of CTSK, there is a significant decrease in the glycocalyx for YSCCC and MCF10AMUC1. So indicating that the CTSK protease can affect the overall glycocalyx thickness in the MUC1 overexpressed cells, or the cell lines or the cell systems that have high MUC1 overexpressions. However, they are not shown the results for the other cell lines in this main figure. So that's all for this video, and that's all for this paper. If you need to understand more about these experiments in detail or about this paper in detail, you can go through the reference that has been provided in the description, or you can also write the comments about this topic in this video that I can try to address it. So that's all for this video. Thank you for your time.